We've now covered the 20 common standard amino acids that typify proteins, but it's important to note that in nature we do find other amino acids besides the 20 common or standard. Some of these amino acids are derivatives of our usual amino acids. Some of them are in fact precursors on the biosynthetic pathways towards those usual amino acids. Some of them are metabolic intermediates that still qualify as an amino acid, even though they're not used to build proteins. Let's talk about some of those unique amino acids that are unusual. So a variety of these, as I said, precursors on anabolic pathways towards the synthesis of our 20 standard amino acids, or also intermediates in biochemical pathways. Let me give you an idea for what I'm talking about here. One of the pathways that we're going to talk about in all its glory is the urea cycle. And in that cycle, there are two amino acids that are formed as intermediates in that pathway. These two amino acids are called ornithine and citrulline. And while they're not used to build proteins, if we look at their structure, we can easily see that they are in fact an amino acid because they have a central carbon, they have a carboxyl group, an amino group, and an R group and a hydrogen atom attached to that central carbon. So by very virtue of that structure, they qualify as being amino acids. We also see some unusual D-amino acids present in nature, and many of you will recognize this from microbiology. Remember talking about in peptidoglycan how we had the sugar derivatives NAM and NAG that formed these chains in peptidoglycan. So I'm showing that chain, and those chains were linked together to one another by something called a tetrapeptide bridge. So tetrapeptide, 4 amino acids all linked together to one another that then linked together these chains of glycans. So this is a tetrapeptide chain here, 4 amino acids linked to another tetrapeptide chain, and I've kind of scrunched my drawing, but another 4 amino acids that would then link together these glycan chains. Well, inside of these amino acid chains are some very unusual D amino acids. We'll talk a bit more about that structure a little later, but suffice it to say that there are some unusual D amino acids present in the cell wall of bacteria. Also, we see some D amino acids in antibiotics, and 2 very familiar antibiotics to all of us are bacitracin and polymyxin. Both are peptide antibiotics, and both of these are containing unusual D-amino acids. So you may have thought, well, Rachel, those don't sound familiar to me, but in fact they are familiar because these are 2 of the 3 ingredients found in Neosporin. So our topical antibiotic ointments. Some common amino acids are modified to produce biologically important amides. Let's look at an example of that. Everyone has heard of, and especially those of you who have allergies, have heard of this molecule. And I'm going to go ahead and draw this molecule, and as I begin to draw it, you might actually start to recognize what this is derived from. Even now, at this point, you may recognize it. We can connect some bonds, and then we've got a chain here. This molecule is histamine. So you know those gnarly allergies, well, we often think about the effects of histamine in the symptoms of allergies. Histamine is, as you can tell, a derivative of the amino acid histidine. So histamine is derived from histidine, our favorite amino acid. We could also draw another of our most sometimes welcome, sometimes unwelcome molecules. This is epinephrine, or also known as adrenaline. Sometimes we get a little too much of it right before an exam, but I'm going to go ahead and draw the structure of adrenaline, or known now by its more modern name of epinephrine. So we'll see a ring structure here, and we'll see double bonds, and maybe even now you're starting to recognize from what amino acid this is derived. And a little more to this structure here because we have a chain on here, a CH bond OH bond CH2 NH2 bond CH3, and there we have it, the structure of epinephrine or adrenaline. So we can see how some very important biological amines are made from, in fact, our common 20 standard amino acids. So we can see that some are, some things are some amino acids are modified, right, and then function in the modified form. We could also talk about how some amino acids get chemically modified to produce things that function uniquely in certain proteins. We're about to talk about collagen, actually, and the structure of collagen. One of the important parts of collagen is that it has a hydroxylated proline and a hydroxylated lysine, which largely impacts the hydrophobicity of those molecules and the ability of collagen to take on water and the kind of compression structure that it can take on. So the fact that that gets modified after incorporation into a polypeptide chain, so even within one of the standard 20 amino acids within a protein can get modified post-translationally. We can also talk about a 21st amino acid. You're like, wait a minute, a 21st amino acid, there's another one. And there is, in fact, and this seems a little strange, Cellino Cysteine. Notice that it looks a lot like cysteine. You think, wow, that's almost cysteine, except for this, there's a selenium that replaces the sulfur. So how does this work? I mean, those of you who understand gene expression will say to me, but Rachel, on an mRNA, there's only so many codons encoding for amino acids, and they encode for certain amino acids, and Cellino Cysteine, I don't remember that ever being in the codon usage table. And in fact, you're very, very right. In fact, Cellino Cysteine is only ever incorporated in rare circumstances when a particular stop codon, UGA, is met. And sometimes it's really crazy, but sometimes that leads to the incorporation in certain systems to a Cellino Cysteine, particularly in the expression of genes that are encoding for dehydrogenase enzymes. So sometimes we see that occur. I just thought it would be fascinating to know that there is a 21st amino acid.
So this next subject is really kind of exciting because we get to put together what we learned previously about weak acids and their titration curves and ionizations with what we have just learned about amino acids. So we're going to look at the fact that amino acids behave as weak acids, and every amino acid, no matter what type, is associated with at least two ionizable groups. So if we look at just quickly reviewing the overall structure of an amino acid with its central carbon, its carboxyl group, its hydrogen, its amino group, and of course its R group. We remember that both the carboxyl group and the amino group are ionizable. So every amino acid, no matter what its R group, is going to be associated with at least two pKa values. And of course we know that for every ionizable group, if it is below its pKa, for example the amino group, then it stays protonated, whereas if it is above the pKa, then we know that the deprotonated form predominates. So if we just briefly write that down, if the pH is less than the pKa, then the protonated form of the group predominates, whereas if the pH is greater than the pKa, then the unprotonated form or the deprotonated form of that group predominates. So every amino acid is associated with at least two pKa values, one for the carboxyl and one for the amino. But of course we could also see situations in which an amino acid would be associated with more than two pKa values because you may have an R group that is also ionizable. So you may be looking at something like aspartate where the R group also has a pKa value that describes it. So it would have a third ionizable group and would have 3 pKa values that would tell you the point at which the group goes from being predominantly protonated below the pKa to being predominantly deprotonated above the pKa. So let's go ahead then and practice a little bit with this. The first example we're going to do together, and we'll talk it through, and then I'm going to have you guys practice a question on your own and see how you do in determining the way in which a titration curve would reflect the different ionizations of an amino acid. Okay, cool. So let's go ahead and get started by looking at a fairly simple amino acid, alanine. We'll draw the titration curve for alanine and talk about the ionizations of both of its ionizable groups. So notice I said both there. Remember that alanine has the simple methyl R group, just simply CH3. So the two ionizable groups are its amino group and its a carboxyl group. Its R group is not ionizable. So this is the structure we would most often associate with alanine, but you guys know that this is the structure that predominates at physiological pH. So it's what predominates at a pH that is below the pKa of the amino group, but above the pKa of the carboxyl group. So this is the zwitterionic form of alanine. So if we will then, let's go down and look at what we've got here in terms of drawing the cationic, the zwitterionic and the anionic form of alanine. We've already successfully drawn the zwitterionic form, right? The form that has the two groups both ionized, both the amino and the carboxyl are ionized. So the zwitterionic form then, being also the uncharged form if you look at that, right? With its CH3 side group. Okay, this is a hydrogen. Okay, so then of course if we went down to really, really low pH, pH is below the first pKa. So the first pKa value here is pK1, which is equal to 2.4. Okay, so at values below that pKa, we know that that pKa refers to the carboxyl group. So that is the pKa for the first ionizable group, the carboxyl group. The pK for the amino group, pK2, is equal to 9.9. So pK1, 2.4 for the carboxyl group, pK2, 9.9 for the amino group. So below the pK for the carboxyl group, we know that the protonated form of that group will predominate. So we would have then the cationic form, we would call it COOH, the protonated form, and NH3+. Right. Now, hang on. Why is that protonated? It's protonated because it hasn't yet passed its pK, and it won't until pH is of 9.9. So the amino group will stay protonated until very alkaline pH is above 9.9. So the hydrogen and of course the R group remain the same. So we say then that this is the cationic form. When you add up the charges on this, you see why. Okay, so there's a plus charge here, but this carboxyl group is neutral, so it's a cationic, right? It carries a +1 charge. We can even write that down, in fact. Let's write that down. Cationic +1 form. Excellent. All right, so hey, check it out. What's going to happen then above the second pK, right? This is the point at which we will finally surpass the pH, going from, for the amino group, going from the point where it is predominantly protonated to the point where it is predominantly deprotonated. So let's go ahead and draw the form that will exist after pK2. So let's write that down. We know then that's the point finally that this amino group loses its acidic, well, it sounds silly to call it an acidic proton, doesn't it? Because in fact, we tend to think of that group as being basic because it doesn't lose its proton until very alkaline pHs. Of course, as always is the case, because they're not ionizable, this R group will remain the same. And of course, we're still far above the pK of the carboxyl group, and of course then it will be deprotonated. So hey, notice it makes sense then that we call this the anionic form, doesn't it? Because when you add up the charges here, the only charge that exists is the negative charge on the carboxyl group. So this is the -1 form. Excellent. So now we see then the 3 different forms of the amino acid alanine that can exist, described by 2 pKs for the 2 ionizable groups, the carboxyl group and the amino group. Okay. At very, very low pHs, below 2.4, the carboxyl group is going to maintain its acidic proton, and as will the amino, and we'll have the cationic form. Now, as you pass this first pK, we go from the cationic form predominating, and at the pKa, of course, those 2 forms will be equal, right? pH equals pKa, the cationic and zwitterionic forms will be present in equal concentration. And actually, let's write that down, because we know at a pH equal to the pKa that these are present in equal concentrations. I'll just use a symbol for concentration there, equal concentrations. So at pK1, these 2 forms are present in equal concentrations. And we might even call this then, what will actually, you guys can think of this. What will happen at this first pKa when cationic and zwitterionic forms, we could say, in fact, if we wanted to, we could call this the acid and this the conjugate base, if you will. What happens at a region where acid and conjugate base are present in equal concentration? What will it resist? Okay, I hope that some of you are yelling at me on the computer or on your iPod right now and saying that this would be the buffering zone, right? This would be the zone in which it resists changes in pH. So we'll see a buffering zone right here. The same would be true at the second pK. Except for at the second pKa, we know that the zwitterionic and the anionic forms would be present in equal concentration. So we would see 2 different forms, and in this case, it's interesting to note this would be the acid and this the conjugate base. And in this case, then, we would see a buffering region around pK2 of 9.9. So we would see a second buffering zone. So the zwitterionic and the anionic forms would be present in equal concentrations. Let's get the word in there this time. And this too would be a buffering zone. Yes, that is a Z. Okay, let's put zone over here. So buffering zone. So there will be 2 buffering zones on this titration curve. There are 2 pKa's. There are then, therefore, 2 plateaus, right? So a plateau at each one of the pKa's. So let's see if we can then go ahead and draw this titration curve, knowing that the first plateau will occur at about 2.4. We draw a dotted line. We might be able to help ourselves in finding that plateau. So dotted line there at the first pK value. So we'll see the first plateau right there at 2.4. So we go here from an area where the protonated form COOH predominates now, deprotonated COO minus predominates. Okay, so right there then at 0.5 equivalents of sodium hydroxide, that is the point of the first pK, pK1. and that is the buffering zone, where you see that it's resisting the change in pH. So if you wanted to buffer something at a pH of around 2.4, you could use the cationic and zwitterionic forms of alanine. I don't know why you'd ever want to buffer at 2.4, but maybe you would. So in any case, then, let's look at the rest of the titration curve, because we know that the second pK value is right at 9.9, so we might just draw a line if we can. I'm sure it won't be perfect, but it'll give us some idea of where to draw that second plateau. Okay, and we'll go ahead and continue up our titration curve. And here we have our predominantly protonated amino and then predominantly deprotonated. Okay, so right there at 1.5, we see we have our second pK value, pK2. Okay, excellent. So we see two buffering regions, two buffering zones, two plateaus, two pK values, two ionizable groups. The number of ionizable groups determines how many plateaus you're going to see, how many pK values there are going to be. So we could think that if you had an amino acid like aspartate or glutamate that has a third ionizable group, you would also see in the titration curve a third plateau. So we would have to add in a whole nother region of buffering, a whole nother plateau in there. Okay, so I want you guys to take a good look at the cationic, zwitterionic, and anionic forms. And we're going to come back to this screen in a moment, but I want to take a little bit of time to look at the regions where cationic and zwitterionic are equal, and likewise where zwitterionic and anionic are equal. All right.
Now, thinking about that, don't let your mind leave that idea of cation equal in concentration to zwitterion, zwitterion equal in concentration to anion at those two pKs. Let's look at that a little bit closer here with a picture. So here we've got it, we've got the cation, right? Hey, what is the overall charge on that species? Well, we call it the cation because it has a charge of +1. Okay, so we've got a charge of +1. What about the zwitterion? What's the overall charge on that puppy? 0. Yeah, 0. Okay, excellent. So if these two species are present in equal concentration to one another, what can you tell me is the net charge? Ooh, I heard a lot of different things. 0.5, right? Yeah. 0.5, right? You know, if you've got a 0 form in equal concentration to a +1 form, overall the net charge is 0.5. So we've got our and in this case you could call this our acid and the conjugate base of that acid equal in concentration. And their overall net charge would be 0.5. And that's what's true at a pH of 2.4. So if you look at your titration curves, look back at that picture, right at that first pK, the overall net charge of alanine is 0.5, 0.5, right? Now looking down and thinking about the pH of 9.9, the second pK, there we're going to see the zwitterion and the anion being present in equal concentration. Now help me again with charge here. We know that the zwitterion is going to carry that overall 0, right? But the anion is -1, correct? Good. Now what would our net charge be here? -0.5, right? Seems, okay, yeah, that makes sense, right? So a -0.5. Where is alanine? Where does it carry a 0 charge overall? If it's overall 0.5 plus at a pH of 2.4 and overall -0.5 at a pH of 9.9, at what point will alanine be 0? Will a solution of alanine be 0?
6.
Oh, some math in the head. Who is that? Oh, good, Troy. Nice work. You said you're right on this. Okay, so how did you get there?
I subtracted 2.4 from 9.9 divided by 2.
You got it. Okay, good. So basically, the point in between the two pKa's is what Troy said. If you find that point in between the two pKa's, you found what we call the pi of alanine. Let me write that down. If you take the first pKa, 2.4, plus the second pKa, 9.9, and you divide that by 2, then you should come up with that value 6, right? That's sort of halfway in between. So that's the pH at which the net charge of alanine is going to be overall 0. We call that the pi. Cool. And you can make this a more general equation, couldn't you, by just saying pK1 plus pK2, and then that would be equation.
Does PI stand for anything?
Yeah, it's isoelectric, that's it.
What is
Thank you. All right. Oh, shall I write that on there, pK1? Now hang on, will this always be true? Not if the R group's ionized, well good. So you may have a situation where you're not averaging the pK1 and 2, pK1 and 3, or something, or 2 and 3 exactly, instead of 1 and 2. Because it depends upon where that zero charge takes place.